Okay, thanks very much, Dave. What we're going to do now is we're kind of switching gears, and there's going to be a series of short presentations, and then we're going to have a break, so you can all stretch your legs, refill your coffee. So we're going to have in setting up this session, I have set a challenge to the researchers who are going to be talking, and that challenge is that they have to present this in a way that you will find interesting, exciting, and, more importantly, understandable. So anybody who gets caught using long and fancy words will be in trouble. So while you're having coffee at the break, the people who use long and fancy words will be having words with me, which will be less fancy and shorter. So what I'm going to do, I'm actually going to start off first. We have to define the nature of the problem in the first place. And once we know where things are taking place, where things are going wrong, we can then try to deliver our therapies to those places. Now, most of what we've been talking about is actually thinking about the brain. And me as a neuroscientist or as a neuropathologist, for a long time ago, I made the decision I would work on things above the neck. So I've always been interested in looking at the brain and finding out which different parts of the brain are affected. And it's very clear that not all the cells in the brain are affected the same way. And we have to be sure that when we are delivering something like gene therapy, we put it in the right part of the brain, some of the parts that are the most sensitive, like those that relay visual information that's coming from the eye. However, from our efforts in treating, for example, mice, we're discovering that we're not curing all of the problems that are actually taking place within the disease. And what we're finding is that there are actually some new, and to some of us, very surprising new targets, new places that we have to think about where we're going to have to deliver our therapies to. And some of those are actually outside of the brain, meaning that we have to start thinking in a whole new direction as to where therapy needs to be delivered to. And we heard a bit about that for the eye, for example. That's a structure which will likely need to be targeted separately. So the first place that we discovered that was actually affected, and this was work that was done by Heymanth from my lab, was that the spinal cord was perhaps surprisingly very severely affected in CLN1 disease. And in actual fact, it's affected before the brain, so it's something that happens quite early in the disease process. And we know that that takes place in the human disease. We know that it takes place in our mice. And if there is pathology there, we know that when we were treating the brains of those mice, it wasn't good enough. And that we also have to treat the spinal cord. And that's important because we've also seen that there is pathology within the spinal cord in every other sort of Batten disease that we've looked at so far. So that includes CLN2, CLN3, CLN6, CLN7, CLN8. Every single form has spinal pathology too, and so we need to think about delivering to that spinal cord. And some of those delivery methods that Dave was talking about, for example, the intrathecal delivery, which is effectively a lumbar puncture, a spinal tap to deliver, would be delivering to the spinal cord too. So that's good news that the cord would be treated if the gene therapy or other therapies are being delivered by that route. And what we did, worked together with Mark Sands, we actually tried doing a double approach, which was treating both the brain and the spinal cord at the same time. And that actually was the most effective combination of way to treat the CLN1 mice that we have done so far. And what we're currently doing now, together with Collaborations Pharmaceuticals, is actually giving the enzyme through the same different routes, both to the brain and to the spinal cord at the same time, and to try and develop that as an effective delivery system for enzyme in CLN1 disease. So that's the spinal cord, but many of the other places that we're actually finding that are affected in these diseases are actually out in the rest of the body. So it's very clearly that these are not simply just a disease of the brain. You know that very well, that your entire child is affected by this disease. So it's not necessarily a surprise to think that there would be effects in other parts of the body, and certainly we're learning very much that that includes the heart, and we're also learning that it involves the guts, very clearly. We certainly also see, we've talked about the central nervous system, which is the brain and the spinal cord, but there are other parts of the nervous system that go out into the rest of our body. That's the peripheral nervous system. There's the autonomic nervous system that helps control our hearts, for example. And there's actually, very surprisingly, also nerve cells within our guts that I'll be talking about in a moment. And it's clear that if there is disease in these places, which our work certainly says there is, this could be causing many of the very debilitating effects of the disease in terms of things like pain, rigidity, muscle wasting, and certainly a lot of the severe bowel problems that your children have. And I think that if we can target those and treat those appropriately, that would actually be a way to improve quality of life to some extent, provided that we're also treating the brain as well. And I think that it's important that we're considering these extra targets. So what are they? So we know that within our sensory, our peripheral nervous system, we're having problems with the cells that actually receive that information. So there are cells that sit alongside the spinal cord that are called the dorsal root ganglia cells. We know that those are dying. We know that they're showing pathology. And we think, as Josh is going to talk about in a subsequent talk, that this is related to different pain thresholds that we see in our mice. We know that those peripheral nerves themselves are actually degenerating, so we're losing the nerve fibers, the processes, and that results in the muscles losing the innervation, the nerve inputs to them, and that may well be what's leading to the muscle wasting that we see in the disease. And we know that this also seems to be leading to problems with how our mice walk, which is something that Heymanth is going to talk about in a moment too. Now we know best of all that all these things are happening in CLN1 disease and will therefore need treating in that form of the disease. But we're also looking at all the other forms of disease too. So we're currently working on both CLN2 and CLN3 disease, looking at exactly the same things happening. Now, another thing that is a whole new thing for us is currently looking at what's been commonly called the second brain. So within our guts, we have something that's called the enteric nervous system, and this came as perhaps a surprise to me, that there are actually more nerve cells in our guts than there are in our spinal cord. And what they're doing there is they're actually doing something very important, which is controlling how our guts actually work, how the flow of food, how we can poop, and all these sorts of things, very importantly, are controlled by groups of nerve cells that sit within our bowels. So we have found in the CLN1 disease that these cells are actually completely wiped out. So if we look here in this picture here, this is a mouse which doesn't have the disease, and in pink, pinky purple are those neurons, and on our CLN1 mouse, they've simply all gone. And the same is true with the glial cells that are supporting them. There's lots of yellow cells here, and they've effectively all gone. So we know that this second brain within the gut seems to be very severely affected in these mice, and that may well be the cause of a whole series of problems that your children are having. Things like reflux, things like abdominal pain, things like that feeling of distension, things like the poor bowel motility or the constipation. So we're currently studying all these things. My kids think it's hilarious that we're now measuring mouse poop, and we're feeding mice coloured dyes and waiting to see them poop red. These are the sorts of things we're now doing in the lab, trying to understand how the disease is affecting the bowel. And of course, we're very keen to try and treat this, and we're also looking at this happening in other types of Batten disease too. So this has opened up a whole new series of questions about how we might treat them. What's the relationship between what's happening in the gut with the brain? Because there's some evidence in things like Parkinson's disease that disease is starting in the guts and spreading up to the brain. We don't know if there's anything like that in Batten disease as yet. We don't know if we're doing gene therapy to the brain, whether that will simply fix all the problems we see elsewhere in the body too. But we're trying to do that. We're trying to look with new types of vectors that will treat either just the periphery or just the brain, or to try and do both. And we're trying to figure out exactly which of the cell types we need to treat by making mice in which we can switch off a gene just in one particular cell type, which we're currently doing by some fancy tricks. So, and we're actually extending all of this, as I say. Most of this we know about in CLN1, but we're extending it to other forms of Batten disease too. So we've heard a lot about mice, and Dave actually mentioned how mice are good. but not that good. And this is because their brains are very small and very simple. They're very smooth, they're very simple. Mice simply want to eat things, not be eaten, and make more mice. They don't do much in the way of thinking. And it's a bit of a challenge. There is a long history of treating mice, which then never actually translates into a proper treatment in people. And that's often because their brains are far too simple for us to actually learn too much from in terms of how to treat them. And that's where a larger animal model, an animal that has a larger and more complicated brain becomes very useful. So larger animal species, and this is, for example, how all the work that was done with biomarin for the development of Brineura was actually done, first testing in mice and then testing in a dog, a naturally occurring dachshund. So there are also sheep models, there's some naturally occurring ones in New Zealand, and we're going to hear a bit more about some pig models as well, which have been engineered. And these larger animals are actually really good for testing how to deliver that therapy and what precise dose might we need to deliver to a much larger species. So we spend a lot of our time chopping those brains up and looking at them. How we do that is quite different to doing it in mice because those brains are more complicated, but we're very good at doing that, and we've been doing this for a number of years now in preparation for doing all these sorts of things. And how we measure and count all of this we have worked out. So we've been doing this with some friends in New Zealand. They've been doing gene therapy, delivering this to their CLN5 sheep. We know that gene therapy spreads very well through their brains. And they published a study last year that said this is actually quite good at stopping the disease progression in the CLN5 sheep and extending their lifespan. And we've actually had the brains from those sheep and are asking what's happened in them. And Kago, who you've seen around, he's involved in doing some of this work. So we know that virus is capable of making the CLN5 protein that's missing. We know that if you look at inflammation within the brain, so this brown stain is showing lots of information in a sheep that's not been treated. That's effectively been prevented in the gene therapy treated sheep, and it's true for both astrocytes and microglia, different parts of the immune system. We know that this gene therapy largely prevents the buildup of fluorescent material. It seems to protect the most vulnerable cell populations, the inhibitory ones, and largely prevent the death of other populations of neurons. So the good news is that for this particular form, it seems that gene therapy works remarkably well in those sheep for a CLN5 form. Now, what we've been doing in preparation for similar studies in CLN1 disease is working together with colleagues in Scotland. That's Tom Wishart, who's at the Roslin Institute in Scotland. It's just outside Edinburgh. And that's where they made Dolly the sheep, who you may well have heard of. They're very good at manipulating sheep and generating new sheep models. So we've been using a new technology, something called CRISPR, CRISPR-Cas9, which you may have heard of a lot about, how this is used for fixing genes. We're actually using that to create the particular disease-causing mutation that's in most of the children with CLN1 disease. And when we do that, we end up with a sheep that has no enzyme, no PPT1 enzyme, which is what's happening in all the children. And we're currently studying those sheep to see how well they copy the human disease and are they like what we've seen in our CLN1 disease mice. We certainly know that they have a disease that looks rather like CLN1 disease and that those sheep go blind, they have problems walking, they have the same sort of sensory problems that I mentioned in the mice. We can use MRI to follow the progression of disease in those mice and see how different selected portions of their brains are affected. And we are currently doing a lot of work on the pathology of those sheep. And here are some pictures of the sheep at the end of the disease. You can see this is a sheep that didn't have the disease, and the brain in the sheep that does have the disease, which is the lower one looked at the outside or at the middle surface, is much, much smaller. The cells within that brain are dying, and the cells that are there are full of the storage material. So we're currently working together with Tom, and we're looking at how their disease progresses over time. That will give us some good landmarks for testing whether the therapies that we have been trying out in mice will also work in those sheep and how we need to scale it up. And those sheep are going to be very useful for us to determining both the best dosing that we need and the best route to deliver in a way that's simply not as possible to do in mice. And so we're interested in trying, obviously, different approaches, both gene therapy or delivery of enzyme, as was done for the development of Brineura. So that's me finished. I have to thank everybody within the lab. This time last year, I had only just moved to St. Louis, to WashU. We're now settled in fully, and I have a great team of people all here. We're assimilating. That was us all going to a cards game. So thank you to them and all of our large number of collaborators. Now, in finishing, I'd like to just mention and announce the date for the next science conference. So this happens every other year, and this is a conference that will be taking place in the fall of 2020. So it's NCL 2020 in St. Louis. So if you make a note of those dates, that's October the 7th through 11th. And we'll be holding that in the Education Center on the campus at WashU, which is a great facility, which is very flexible and set up both for a big conference, but also breakout rooms and sessions. And when I organized the equivalent conference in London in 2012, we had a whole series of sessions that were specifically designed for parents, and one of my main aims is to have science to be accessible for all of you. So we're working on the structure of that program, but it will certainly involve very similar things. People from our labs will be at the end of each day decoding what we did. Also, because it's happening to be taking place on campus where we are, it's in the building next to our lab, we shall be arranging before the meeting the chance to come and actually see our labs and see what we do and see some of the things, and I hope to demystify what it is that scientists do when they're in their labs wearing white coats. So you will all be very welcome to come. Those are the dates. We're busy organizing the conference and raising the money to make that conference happen, but you will all be extremely welcome to come and attend if you can make it there. So that's me finished. Thank you for listening, and I'm just going to now hand on. Because each of the first three talks before our break are on very similar subjects, we're going to go through those talks and then quickly answer some questions before we have a break. And I'm going to hand over to Hemant from my lab, who will tell you a bit more about the detail about the work he did investigating the spinal cord and the early events that are taking place in CLN1 disease mice. Thank you, Hemant.
Thank you, John. So I apologize for those of you who've been here since the morning, because you're going to have to listen to more of me. I'm Hemant Navagul. I work with John Cooper, and as John kind of introduced you, I'm here to talk about work that I've been doing in his lab about studying the disease progression in the spinal cord in CLN1 disease. So we all know what CLN1 disease is. It's in classically the infantile form, and we know that it severely affects the brain. However, we know that treating only the brain has proven to be insufficient in getting an efficient therapy. So we thought there must be problems elsewhere that we haven't addressed despite these therapies that have been targeted the brain. And one thing that we hit upon was the spinal cord. Now, the spinal cord acts as sort of like an information highway, taking information from the outside of the body up to the brain in terms of the sensory information, as well as information from the brain to the outside of the body to help us move, the motor information. We didn't have much human samples because they don't usually collect them, and we were very fortunate that our collaborators in Finland had a sample from the 1970s lying around, and we had just one. But what was surprising is when we did look at it, we found that there was significant, I don't know if you can see that pointer. Yeah. So we saw that there was significant amounts of disease symptoms in the spinal cord in this one sample that we had, and that kind of gave us a clue that, hey, something might be happening there. So we decided to tackle this problem by then looking at it in mice, as we normally do. And we have a mouse model of this disease where the gene has been knocked out, and what that effectively means is we've removed that CLN1 gene out of this mouse. And for the most part, work in John's lab and many other labs over the years has shown that that mimics the progression of disease in the humans quite well. And where a normal mouse lives about 2 years, our mice live about 8 months. What is interesting, though, is that we knew for a long time with age, they have a lot of difficulty moving, and they have this classical wobbling gait, so they just kind of walk funny and they struggle to move around with age. And we thought, hey, we know that the spinal cord might be affected. Let's have a look and see what happens. So we got this fancy bit of equipment, which is appropriately called a catwalk for mice, right? And basically what it is a little tunnel that you can see in this image here, that the mice walks from one end to the other. However, it has a glass plate floor and a video camera underneath it. And so you can get a video of the mouse running against a red background. So I'm just going to walk you through what that actually looks like in a video that I hope will work. So when you actually start the video, you can see that mouse running from one end to another, and you can see those footprints being picked up automatically. And what that does is it tracks not just where each paw print is, but how long the mouse spends on each paw. What that also means is we can actually see in real time which bits of the paw, so whether they're walking on their toes or whether they're walking on their hind paws, and how much pressure they're putting on them as well. And we can classify them as, right front, right hind, left hind, and so on. You can also see, and this is just showing the amount of pressure they put on each one. The last thing we can also do, mice walk in consistent patterns, so that when you put your, we all walk with two legs, so we just left, right, but they go right front, left hind, left front, right hind, like a cross motion. And we can track how consistently they're walking as well. So you can see it's quite a powerful tool. It's not just all fun and games to call it a catwalk and make them model their walking. So we get a lot of information out of this. Things like how fast they walk, how many steps they're taking per second, how big their steps are. And all of this gives us clues as to how well they're able to move, right? So what did we actually find? And I apologize that this slide has a lot of ugly graphs. What I'm going to direct you and orient you to this is basically all these graphs are the ages in months on the lower axis. So every point there is 1 month. So how they're doing it, 1 month, 2 month, 3 month, up until 7 months, and how things change. Now, with mice with any disease that have difficulty walking, you'd expect them to start off normally and then just decline. And that's pretty much what we see across the board. Across most parameters, they kind of decline compared to, so the mutant mice are the red ones, and the wild type or normal mice are the blue ones. And you can see that the red lines always tend to go down towards the end, and that means they're not able to walk. However, what was really surprising to us was that really early on across, the top two graphs and the bottom left graph, is that if you look right at the early point at 2 months, when we know that there's nothing happening in the brain in terms of disease pathology, they seem to be walking faster, they seem to take bigger steps, they seem to take more number of steps per second, and then that just made us go, that's interesting. And we didn't really know what to make of it, so we thought, let's actually look at the spinal cord and see what might be happening there, right? So, we know that in the brain, there are lots of features of brain disease that are characteristic of CLN1. The brains become smaller, and as both Dave and John mentioned, there are these supporting cells that are associated with immune responses in this disease, the astrocytes and microglia, and these become activated. And that's a bunch of functional changes, so their function changes, there are a lot of chemical changes, but what we can see visually is that they actually become, they stain bigger, and they become more what we call activated. There's also accumulation of the storage material, and there's loss of neurons or nerve cells. So we know that these happen at specific time points in the brain. At 3 months, all of these stuff happen, and at 5 months, you see accumulation of storage material and nerve loss. So our question was, does that happen to the cord? And if it does, when does it happen? But also, is there anything different about the spinal cord, right? So we're tackling one of these at a time. The first thing we looked at is volume changes, or whether we know that the brain shrinks over time. Is the same thing happening to the cord? So the cord is made up of 2 bits, and you might have heard of these terms, white matter and gray matter, right? So gray matter is all the cell bodies of these nerve cells, whereas white matter is all the connective bits, what we call axons and dendrites, which is all this stuff that stick out and make connections with each other, right? So we measured the differential parts. We measured the total volume, and we measured the volume of the gray matter, which is this butterfly-shaped things in the inside of that cord. And by subtracting these volumes, we got the white matter volume. And again, I apologize for the graphs, but basically you can see with the blue lines being normalized, as early as 2 months, the mutant mice don't really meet those volumes, meaning they start off really early in disease, just not being the same size of the spinal cords as wild-type mice. So that was volume changes. The second thing we looked at is, do neurons die in the cord, right? Because nobody knew the answer to that. And the answer was, yes, they do. And what was also surprising, especially in the graph on the left, is that happens really early in disease. What I mean to stress here is these neurons are dying in the spinal cord much before, nearly 2 months before you see neurons dying in the brain, right? And that's significant. That was surprising because with disease like this, you might expect that cells die eventually everywhere. But it's surprising to see things happening so quickly in another region. The next thing we looked at was the accumulation of storage material, and I hope these pictographs are representative. So on the left, you have what is a 7-month-old normal mouse, and then you have the PPT1 or INCL mouse at 1, 3, 5, and 7 months. And you can see as early as 3 months, there is significant accumulation of the storage material. There is microglial activation that we talked about in terms of the immune response and astrocytosis. And you can see by 7 months, that's just filled up the entire cord. So all of this was really interesting, but we asked, is there stuff happening that we don't know about that isn't similar to the brain? We know that the white matter isn't significantly affected in the brain. But when we looked at the white matter in the spinal cord, we saw that there were significant amounts of changes that are associated with disease from other ideas, that we've learned from other diseases. Those hallmarks present in the spinal cords of the INCL mouse as well. So we now have a different picture of all these things that are supposed to be happening in the brain. We know that all of these things happen at these various time points in the brain, and all the same features show up in the spinal cord, but much earlier, like they phase shifted earlier. Also, we know that there are some features, like white matter pathology, that are happening in the spinal cord that aren't really happening in the brain. So the next bit, and John mentioned this briefly in his talk as well, is something we call the dorsal root ganglion. So this is a section of the spinal cord that we see here. But all the information that comes from the periphery, from your hands, from feet, from your skin, all of that stuff, first relays in these nerves, in this little ball of neurons just outside the spinal cord before they then relay to the neurons in the spinal cord. So it's important to also understand, if you're talking about the sensory system, what's happening there too. And we're starting to ask that question, and we're able to see that yes, there is a lot of storage material, there is an immune response, and there's a change of the supporting cells as well. So again, showing that this area is also affected. So now we know what's affected, the spinal cord, the DRG. We know when they're affected, much earlier than the brain. The big question is, why? Why is the spinal cord any different from the brain, right? So to begin answering that question, we use this technique called proteomic analysis. And some of you might have heard a lot of names like genomics and transcriptomics and all these fancy terms. Proteomics is basically a big picture view of a lot of proteins in the tissue. And we know how some of these relate and how some bunches of these proteins work together for a certain mechanism. So we look at this big picture view and go, all right, if you knock one protein out, in our case CLN1, what happens to all the other proteins? What happens to all the other mechanisms? So to try and understand which mechanisms might be affected differently in the spinal cord that aren't affected in the brain, right? So now we get to some really messy pictures. They do look pretty from like a distance, just little wheels, right? Also, it's nice for my boss because I can say, look, data. Basically, each of these wheels represents, each of those points on these wheels represents a protein. And we know, like if you Google search them, this protein is associated with this process in a cell. There's all these papers that have been published over many, many years. So you can start grouping them as to which one's associated with which processes. And every time they go red, it means that the levels of that protein are higher in the disease than in the normal, and green, they're lower in the disease than normal. And the more numbers of these proteins that are changed in a particular process, the more that process is affected. Now, the first little wheel or cycle there is the lysosomal defects, which is a good thing because we're studying a lysosomal disease. So you see all these other lysosomal proteins go up and down. So you're looking at a real effect. Interestingly, things that we don't see in the brain started showing up. Things like metabolism of fats or lipid metabolism. Things like myelination, which is the fat covering of the connections of these neurons, seem to be affected. Proteins associated with the development of the sensory system seem to be affected. So now we start to get clues about what's happening different in the spinal cord compared to the brain. We then compared that to what's happening in the brain, right? So again, ugly heat maps, but basically, every time each of these lines is a process. And the darker the color, the more number of proteins are affected in that process. And the lighter the color, the less that process is affected. And the left column, SC, is the spinal cord, and the right column is the cortex. So you can see, generally, there are a bunch of processes that are more affected in the spinal cord than they are in the cortex. And the same goes for things associated with particular diseases. So from this, we can start to make observations like there is a much earlier spinal cord immune response as compared to the brain. There are more degenerative changes seen in the spinal cord and things associated with myelin defects, which we don't see in the cortex. We then decided to become a little more focused and look at this immune response in particular. And what we did was we looked at a bunch of different molecules that are associated with immune responses. We looked at them at 1, 2, and 3 months when we know that these early changes are happening, which is what each of these little things is. And I'm not going to draw your attention to any of these, but this is basically just to draw some red lines and say, look, a bunch of stuff is changing at 2 months, right? And that's the technical term, I think, a bunch of stuff, right? But all of this is more evidence that things are happening in the spinal cord much earlier than we thought were happening. So we have all this information. What can we do with it? So in working with our collaborators at WashU in Mark Sands' lab, we published a paper a couple of years ago which John briefly mentioned, where we treated with gene therapy both the brain and the spinal cord together, right? So we studied the lifespan of these mice. Now in red, we have the mice that are the mutants, and in blue you have the normal mice, which seem to live past 20 weeks. When we treated just the cord, you can see that there was a slight improvement. That's the green line there. And when you treated the brain only, you can see a slight improvement that's a bit better. That's the yellow line there. But when we treated both of them, you can see that there's this massive shift to the right, where these mice live so much longer. Not only that, in the graph to the right that says rotarod, which is another way we just measure how well mice move, we can see that there's a similar pattern where the combined therapy means that these mice move much better or closer to the unaffected mice. So we know that all of this is great. Like, we know the spinal cord is affected, great. But the important thing is, by treating it, it has a real effect clinically. So just to summarize, what does all of this mean? Why should you even bother with this talk? It's all about the timing of the therapy and the targeting of the therapies. Early pathology means that you redefine that window in which you can get the best therapeutic effect. Novel regions of pathology means that you don't just target your therapies to one area, but you need to start thinking about all the other areas. And as John mentioned, now we're going past the spinal cord and looking at things like, the enteric nervous system and the peripheral nervous system. And we need to start asking these questions, not just in CLN1, but across all the NCLs. And also, learning about these new features of the disease not only allow us to provide better tools for diagnosing the disease early, but also by learning the different mechanisms that are involved, possibly finding new therapeutic options, new things we can treat, right? So to continue this, my colleague Josh is going to talk about some more behavior we've done in these mice to try and understand more about how they react early in this disease. We're also working on finding out pathology in these connected regions, like John's talked about, in the dorsal root ganglia, the peripheral nervous system, and the enteric system. And I just put this up there because I like these pictures. We're doing these weird, complicated, for me, cluster analyses to try and understand how these proteins interact and which mechanisms are specifically involved in each part of the nervous system. So with that, I hope that was all right. I'd like to thank everybody in the lab and our collaborators, especially at WashU and Tom Wishat at the Roslin, and thank you.
The pathology is showing up earlier than we initially expected, and we're seeing it in the spine and the peripheral system. What are the consequences of that, behaviorally speaking? What we already know about disease or about behavioral symptoms in the PPT mouse is that they are largely restricted to the sensory and the motor domains. So we're talking about things here like motor coordination, muscle movements, the ability to balance. And these mice, they have relatively intact cognitive and memory abilities for most of their lives, and they have relatively intact emotional abilities. They simply don't look much different from their normal or wild type counterparts up until very late in disease. And as Hemant mentioned, these mice die early, around 8 months, so they don't start to look very severe until 6.5 to 7 months. As has already been mentioned, we up until recently thought that this disease showed up in the brain at 3 months of age, and you've seen 2 great talks detailing how we have now stepped it back even further, and we're seeing significant disease burden in the spinal cord and the periphery as early as 1 month of age. And to fill in some of the gaps of knowledge here, Hemant talked about some of the walking symptoms that we've seen on the catwalk. And I'm an even more basic scientist. I put mice in mazes, so I think to everyone's benefit here, hopefully this stuff is very easy to understand. I ran 3 different tests, and one of them is a locomotor activity test to see how the mouse explores an open field. The other is what I'm going to call a walk initiation task, and I'll describe that in a little bit more detail as we go along. And then what I think is maybe the most interesting task is this touch sensitivity test. So how does a mouse react to a very mildly painful stimulus? And I think that gets to these dorsal root ganglia that we're seeing kind of disappear or die very early on in disease. I'm going to go through these one by one, and we tested these mice. We were focusing on early time points, so 1 month, 2 months, and 3 months. This is a very simple, straightforward test, the locomotor activity test. You put a single mouse in a large open chamber and allow them to explore for 1 hour of time. You can measure their horizontal movements, so how far they travel. You can also track, as you see the mouse on the screen there, their vertical rears. So like your cat or dog at home might do, they rear up on their haunches. This is a really easy way for them to explore the environment around them. We're able to split this data into time versus in the center versus the periphery, and that can sometimes be useful if we suspect that we have a particularly bold or fearful mouse. I'm not going to talk about those zones today, but I wanted you to see just how we do this test. And I'm only going to talk about the first thing, which is distance traveled. We see that at each of these 3 early time points, 1 month, 2 months, and 3 months, our PPT mice, which are now in blue, they travel a significantly shorter distance compared to their wild-type counterparts. And that's at 1, 2, and 3 months of time. But when we switch over to those vertical rears, again we see a deficit in the number of rears just at 1 month of age, but the PPT mice catch up at 2 months or at 3 months. We don't have a good explanation for that right now. It's just an interesting tidbit that we'll try to tie together as we move forward. Now, this second test is the walk initiation task, and it's simply you put a mouse in the middle of a square on a wide open table in the middle of a wide open room. And this is a very vulnerable situation for a mouse. They don't like being out in the open. If you think you can extrapolate that to a mouse in the wild, needs to be hidden from predators. We just measure how long it takes the mouse to escape that square, and it's a simple and very crude way to measure the mouse, both their ability and their willingness to initiate movement, to either explore the novel open area around them or to escape a scary situation. And we don't see any differences in walking initiation at 1 month or 2 months, but at 3 months of age, our wild-type mice, those normal mice, they're taking longer to leave that square, whereas our PPT mice, our CLN1 model, they're still scurrying right out of that square. And another useful way to look at this data is to put all of those data points on one graph so we can look at how that progresses over time. When we do that, we see that the wild-type mice, they show this nice linear pattern. And that's not a pattern that we see in our PPT mice. This could be an indication that our wild-type mice are getting used to the task over time. They understand there's not a threat here. Maybe they're remembering the task better, whereas our PPT mice are not. But there's also a possibility that as our PPT mice age, they become hyper-reactive to handling. So when you pick them up out of the cage and place them in the middle of the square, perhaps that's exacerbating their response, and they're just going to run away from the stimulus wherever they are. And then the last test I mentioned, it's a touch sensitivity test, and really it measures the mouse's sensitivity to a tactile or a physical stimulus. I'm talking about a literal poke here, so I'm prodding the mouse with a stick. And we put them in a plastic cage, and we have access. They're on a mesh screen. We let them habituate, get very calm, and then we use what are called von Frey filaments. And that's very simply, it's like a very thin wire or a hair that's kind of attached to the end of a pen. So if this is a pen, you've got a little wire sticking up. And we have a series of 8 of these, and they start very thin, exerting very light force on the hind paw of the mouse, and we scale then up through 8 different ones, and that 8th one is very thick and it exerts a lot of force. We're simply measuring the mouse's reaction to these pokes. It's a model that is commonly used in testing how spinal cord injury or peripheral nerve injury affects just how mechanical your sensitivity to a touch stimulus. So overall, there's no differences in sensitivity between our PPT mice and our wild-type mice at 1 month and 2 months of age. But then we see a significant difference at 3 months of age. The PPT mice are actually what we would call hyposensitive. So they're less sensitive to all of the stimuli. They're not feeling that touch as well as the wild-type mice at 3 months of age. But if we look a little bit closer at the 1 month time point, although there's not an overall difference, we're seeing a trend at least in 2 different points to a hypersensitivity, especially to those very light stimulus. So I told you we start with very light and we build up to very strong. At 2 of these points, they're showing an exacerbated reaction to very light stimulus at a very early time point that we thought nothing was happening up until recently. And now we know the spine is diseased and the peripheral nervous system is diseased. Okay, so we've got these 3 very crude tests. We take it all together, and what does it mean? I'm a behavioral neuroscientist, which means all I do is put mice in mazes. I could talk for hours on end about what I think is happening here, but when we put it in the context of Batten disease, really these are indicators that we can detect the disease much earlier than we initially thought, as early as 1 month of age. And what we think it means is that what John and Heymanth just told you about all of this spinal cord and peripheral pathology is likely at least partially responsible for some of these motor and sensory manifestations. This kind of research becomes really important for a couple of reasons. One is that we want to make sure that we understand the disease in full so that we can create what we call biomarkers. How do we measure how the disease progresses? How do we measure how effective a treatment is? But it also gets to the main point that it's very important that we are modeling the human disease as closely as possible in our mouse. And I have a poster with some of this data on it. I would love to speak with any clinicians or families who have had experience. They notice they think their child or their patient has shown a hyper or a hyposensitivity to tactile or touch or manual stimulus. Come by and talk about it. We're trying to compile and understand everything that's going on here. I know we're slightly over time, and I'm between you and hot coffee. I will only take 1 or 2 minutes. I want to give you a quick update on a project that I proposed in Pittsburgh at this very meeting in 2017. And I wanted to simply ask the question, does cannabidiol show any positive effect in lessening or stopping seizures in the CLN1 mouse model? Just a little bit of background. If you haven't heard of cannabidiol, it comes from the marijuana plant. It's not the part that gets you high. It's not psychoactive. We all have cells in our body that bind to the chemicals in marijuana, the cannabidiol, the THC. And I think everyone in this room has likely heard at least some anecdotal evidence that cannabidiol in particular has shown benefit in a litany of medical conditions. If you've been on the internet, you've seen this. And there are even a few clinical trials showing some limited but promising results in children who have seizure disorders. And over the last 2 years, I've really just been trying very hard to answer the question of whether CBD or cannabidiol will be helpful in the CLN1 mouse. Specifically, will it lessen or get rid of their seizures? And we have great collaborators in the neurology department at WashU. We can measure the brain activity, electrical activity, for extended periods of time. We've gone through a lot of trial and error, and frankly, in retrospect, what feel like failures, and to try to find a CBD regimen that we think will work. We chose one and we moved forward with it. We took our mice at 6.5 months of age, which is a time at which we know they have seizures, and we think they're healthy enough that we could benefit those seizures. But unfortunately, the data hasn't borne it out yet. These are very small numbers. We're having to treat the mice multiple times a day, so we're having to handle them multiple times a day. And as you can see, our vehicle or our placebo is actually outperforming the CBD. This isn't a death knell to our research. We suspect that the problem with our treatment regimen is that we're handling the mice multiple times a day. I've worked with these mice for 8 years, and I can tell you anecdotally, they are as they age and the disease progresses, they are very reactive to handling. They don't like being picked up, unlike their 1, 2, 3 month counterparts. They get very stiff. They show almost a seizure-like behavior just being handled. So we know that handling these mice multiple times a day probably not beneficial to their health. So we had to take a step back and think, what's a way that we can get this drug into the mice without actually picking them up? And on a suggestion from John Cooper, we spoke with Dr. Stephanie Hughes at the University of Otago in New Zealand, who has taken this innovative method. We took a scientific approach, and we did the next logical thing. We're putting the drugs into tiny little cubes of raspberry Jell-O. And what I can tell you is that even if they won't show you on camera, they're not great performers. They won't eat it on camera. But if you give them a cube for a day, they will eat the whole thing. You can give them a cube the next day. They will eat the whole thing. So I can now put the drug into little cubes of raspberry Jell-O. They will eat it right up, and I can get all that drug into their system without ever having to touch them, which is way better for me and way better for them. All of that to say, we're now well on the road to exposing these mice to CBD over long periods of time, over short periods of time. What I would love to do is come back to this meeting in Cleveland and tell you, look, we've got a basic science answer to, do we think CBD is going to help seizures and CLN1 disorder, at least in these mice, or do we not? And it's going to come by way of Jell-O. So I want to thank my collaborators, but I also want to thank especially the BDSRA started funding me on this project 2 years ago, and specifically the BDSRA Australia and Drew's Hope were so generous in giving me some money to get this started, which we were able to parlay into a larger NIH grant. So we're now funded to do this work, and I want to see you guys in Cleveland. I want to give you some good answers. Thank you so much. Go get some warm coffee and warm up.
Before we start that session, while people are still coming in, I'd also like to highlight that after lunch, there's actually a research open session. So this will be in the same room where the trivia quiz took place last night. So that's on the A level, behind where the breakfasts are served. And that's where we've asked a whole series of researchers, including quite a lot who aren't at the meeting here, to put together a poster, so a large, big piece of paper, describing a lot of the work that they're doing. And those of us researchers who are there, and people from some of the companies developing the therapies and setting up the trials, will all be together in that room, and it will be your opportunity to come and grill us and ask us any and every question that you wish to fire at us. And I'd also like to say is that we're here for the whole conference. If you see us around, we'd like to think we're approachable. You can typically find us. Just come ask us. Don't be shy. Come and ask any question. And there is no such thing as a dumb question. There's just a question you don't know the answer to yet. So don't be shy. Please come and ask. Okay, so it's a great pleasure to introduce an old friend, Jill Weimer. So Jill's from Sanford Research, and she's going to talk about the work that she's been doing there. Thank you very much, Jill.
Let's see if I can hold a coffee cup to keep my hands warm and a clicker at the same time. So I'm actually going to pick up pretty much where Josh left off, and kind of serve as the intermediary between the clinical team from Rochester on some of the clinical progress that we've made over the last couple of years. Before I do that, I do want to kind of just give a disclaimer that some of you guys are aware of. John mentioned that I run a research lab in Sioux Falls, South Dakota at Sanford Research. But as of about a month and a half ago, I also have a second job working with Amicus Therapeutics on the gene therapy programs that our lab really spearheaded the preclinical work on. So I'm going to focus today on the work that we're doing at Sanford and not really focus on the gene therapy programs, but I will highlight those as kind of a part of this program. So as Josh mentioned, one of the things as basic scientists that I think sometimes we start to lose sight of is how this actually relates to your children. How does this relate to Batten disease kids? We get so down in the weeds in understanding the biology of what these proteins are doing, working with mouse models that are not really good models of your children, that sometimes I think we need to step back, and we have fortunately folks like John Mink who constantly remind us that mice are not people, and we really need to look at these through the lens of the patient and understand what's going on in the patient and try to model that in the diseases that we're studying. This is a slide that should be familiar when Dave said he stole all my slides. I tried to go through, make all new slides, but over the last probably 5 years, our lab has had a heavy focus on therapy development and tried to step back and look at what are the needs in the field, where are the gaps that we are finding in making that step to translation and understanding what's going on in human patients. And today I'm just going to touch on some of those programs and sort of areas that we're trying to fill in the gaps and start to answer some of these questions. When we think about therapies, as Dave mentioned, he kind of went through what's going on in each one of these spaces, but as you guys know, it's probably not going to be one treatment or one drug that's going to cure Batten disease. It's likely going to be a combination of these, and as we have drugs that are hitting clinical trials and hitting the market, like the enzyme replacement for CLN2, as basic scientists and translational scientists, we have to start thinking about how we're shifting the natural history of Batten disease, because now CLN2 patients, many of them have been on ERT. That's going to shift what we know about that disease, and how do we start to work that into future drug screening? And then also, essentially, every time that we do a drug screening, we have to do it in the context for CLN2 of patients or mice or cells that might be receiving the enzyme replacement and those that aren't, which really kind of starts to complicate and add many layers to even just the basic science that we're doing. I don't think I need to remind this group that all of these diseases are genetic, and I think that we've seen a lot from Dave's presentation and what's going on in the field, that genetic approaches like gene therapy, antisense oligo, exon skipping, read-through compounds can really target some of those underlying genetic approaches, but many of the other drugs like the enzyme replacements, the small molecules, and many of those other drugs can really kind of help to aid in slowing the disease progression, and so we have to start thinking about tackling those together. And ultimately, what we want to be able to do is take this, what hopefully soon will be an antiquated model for drug development, in which when we start sort of screening drugs in the lab, that it might take 5 to 7 years to get something to market, and really turn this on its head so that we're getting effective drugs in place for your kids in their lifetime, and are getting those screened into market. So I want to kind of just step back from the high level, like I mentioned, and think about or discuss some of the questions that our lab has been looking at and how we actually think about therapy development and testing. And the first one that I'll put up, this is the only thing that I'm going to say about sort of the gene therapy programs and the approaches that we take. Dave mentioned that we, with many of the forms of Batten disease, we don't know everything about the biology. And as of about 5 years ago, one of the sort of central dogma in the field was that genetic approaches like gene therapy would not be effective in treating disorders like CLN3, CLN6, and CLN8, because unlike CLN2 or CLN1 that are secreted enzymes, these are very complicated proteins. They loop through the membrane of organelles inside of the cell, and there was a sort of a central thought in the field that overexpressing these would become toxic and actually kill neurons in the brain. And it was actually work that Tammy Killian did where she really said there's no evidence of this in the field. We don't know if this is true. We're kind of basing our inability to develop effective treatments from kind of going on this hypothesis where there's actually no science to support this. And so she developed in the lab an AAV9 CLN3 gene therapy that then she tested in a mouse model of CLN3 Batten disease and showed that overexpressing CLN3 was not toxic to these neurons, that she could get it to fold correctly in the membrane and be expressed. And that's where we really jumped into this and looked at the Batten disease as a whole and said, with sort of that central idea, what are the forms of Batten disease that kind of live up to that? CLN3, 6, and 8 are all transmembrane. They all kind of had suffered that there were no treatments that were effectively delaying the onset of these diseases. And so we partnered with a team at Nationwide Children's Hospital to develop AAV9-based gene therapies for these forms of Batten disease. And as Dave mentioned, the baton has been passed now to Amicus. These are in clinical trial at Nationwide in partnership with Amicus for CLN6 and CLN3. So I think the first lesson for us was we have to think of these as individual diseases, but we can learn and look at the biology and actually design effective treatments that are kind of predicated on our knowledge that we do know about the disease so that we can move that needle faster. The second piece of this is that even when you look within a subtype of Batten disease, so let's say CLN3, that not every form of CLN3 is the same. Kids have different mutations, they have different environmental exposures, there are differences between males and females. The disease progression can be different even between siblings with identical mutations. The assays that we screen in the lab or the clinic may not be relevant to the disease itself. So how can we actually step back and start to model this really in the age of thinking about combinatorial treatments, kids that are receiving ERT or gene therapy? How do we actually start to test treatments in those kids? And so what we did is step back and take a sort of more holistic approach in how we model things in the lab. And so we moved to a system where we're using advanced microscopy and machine learning to use a tool that's called high content screening. And so this is a way for us to take human patient cell lines, induce pluripotent stem cells, or neurons that we make from animal models of Batten disease. And many of you have heard of high throughput screening, which is the concept where you're screening thousands of drugs at once and just looking at one assay. This is kind of the reverse of that, where you're focused in on one or two drugs, a couple of drugs in combination. But now we can set up assays where we maybe have 5, 6, 7 different patient cell lines with different mutations from different sects of children that were isolated at different stages of the disease, and screen multiple different drugs in one dish and start to look at a number of different parameters that are really tied to the biology. And really, as John said, kind of start to automate these processes for whether it's cell-based assays or pathology in animals, and really start to get at the root of these problems. So this is sort of just an example of a scenario in our lab where we might at any given time be looking at a number of different drugs. So we have an assay where we're looking at neuroprotective compounds, anti-inflammatory, or neuroinflammatory agents, modulators of autophagy or lysosomal biogenesis, put those into our assays where we're screening different sets of patient cell lines. And we can set these assays up too, where maybe the same neuroprotective and autophagy drugs might be beneficial for 2 or 3 different forms of Batten disease. So we can be screening these simultaneously and really honing in on the key drugs that we want to screen before we would actually ever even go into any mouse models. The second sort of high-level piece that we had struggled with, and I think you heard this in John and Hemanth's talk, and really in Josh's, is how do we actually take the information that we see in human patients that Rochester and Nationwide are collecting in these clinical trials and really mirror that in our animals? Because for us, historically, the way that we actually show that a disease is working in a mouse is we sacrifice it and collect its brain. And we can't really do that in people. So we need to look at the tools that we have to use in the clinic, and how do we actually start to look at that data as a way to hone in the longitudinal progression of the disease. But as Josh and Hemanth mentioned, really start to get the behavioral phenotypes that we're seeing tracked back to what we're seeing and mirroring the pathology. So I'm just going to draw your attention to the upper right-hand corner. And so this is sort of some of the pathology that John, Hemanth, and Josh talked about that we see in different forms of Batten disease. So this just gives you a table that talks about CLN 3, 6, and 8. And for all of these in our lab, we can see autofluorescent storage material in these models by 1 to 2 months. In the CLN 2 mouse model that Josh mentioned, we actually see storage material by 2 weeks of age. We see these reactive microglia kind of follow in suit a few months later, but strangely, even though the brain is degenerating, and this is probably the case with your children as well, we don't actually see any symptoms of the disease or behavioral changes or vision loss until 6, 7, 8 months of age. And when we think about this with kids, we want to be able to learn as much as we can and know as quickly as possible when their disease is starting and have good tools that we can track this longitudinally in the clinic so we can watch whether a therapy is going to be effective. And we often refer to these as biomarkers of the disease, so ways that we can track them. And I think scientists have tried to think simplistically about biomarkers. So I always use the analogy that we're often looking for a needle in a haystack, and we're having trouble in the Batten disease realm finding that needle in a haystack. So the approach that our lab has taken is actually to look at the whole haystack. So why not step back and actually look at all the data that we're collecting in the clinic, mirror that in mice or in some of these large animal models that John mentioned, and start to develop a biomarker scoring system that's really reliant on that data. And so what we have done in partnership with a team at Charles Rivers is step back and say, what are the things that clinically we collect on Batten disease patients? And those are shown on the right. And so we look at things like MRI, advanced imaging technology like diffuser tensor imaging, spectroscopy. Hemanth mentioned this catwalk system that they use for looking at motor movement. We use something called kinematic gait analysis that is very similar, but this actually, instead of just looking at the paw prints as the mouse walks down this walkway, it looks at movement in 3 dimensions, which is much more akin to the motor rating scale that John Mink's team has developed, and looking at things like PET. We can layer in here also ERGs, OCT, EEGs, a lot of the things that your kids are getting data collected on in the clinic. So we took our CLN6 mice as a starting point, and we started looking at different time points and collected all this data. And then through a series of machine learning, we're able to condense this down. We're essentially, Hemanth showed you these heat maps, and these heat maps are essentially at each stage for each of the parameters. So if we take all the data we collected from MRI, DTI, SPEC, kinematic gait analysis, and PET, we ended up with 144 variables. And then we essentially tell a computer to go through that data and pick out for us in each one of those clinical modalities what were the 3 most important variables. And then we condense that down even further using a contrastive principal component analysis and are able to come up with these biomarker scoring system where now we're picking up phenotypes in these mice 2 months before we were ever picking them up before. And I say 2 months before, this is actually the earliest we've looked. I think that particularly in the females for CLN6, if we back it up even a couple more months, we'd be picking up phenotypes in these mice even before that. So now the sort of next step in this side of things is, can we actually take this, take the data that's actually being collected in the clinic, run the same sort of modeling and machine learning, and come up with a scoring system in the patient. The other side of this that our lab is really focused on is moving away from imaging technology in which a mouse or a pig or a human would have to be sedated and undergo anesthesia, and really focus on things like EEG, nerve conduction, biofluids, readouts that we can get from biofluids, whether it's blood or urine, and coming up with the same sort of modeling so that we could have a biomarker scoring system. And we really believe that once we actually have the mathematical modeling in place, that this actually could be applied across all forms of Batten disease to be used as a biomarker in the clinic. And then just in the last 2 slides, I'll mention John had said that we would hear a little bit about large animal models of Batten disease. So in collaboration with David Pierce's team, over the last couple of years, we've been developing large animal models for CLN2 and CLN3. And so this is just the sort of schematic for how we made those CLN3 pigs. They have the common delta 7-8 truncation, and we're in the process of showing at the pathological level, this really mirrors what we see in other animal models and in human patients with Batten disease. We do a lot of behavior testing with these. Pigs are not easy to work with for behavior testing, but we do a lot of walkway analysis, looking at gait, looking at motor learning in these animals, and really, again, modeling a lot of what we look at in human patients. And just as sort of a proof that these animals, these large animal models, are much better models of Batten disease than the mouse and rodent models that we currently use. You may recall that Dave Pierce mentioned that in CLN3 alone, these animals, the mouse models that we use for drug testing, do not model what we see in kids. In fact, they never have vision loss and they never succumb to premature death. But in our pig models, I'm showing you ERGs from 2- and 3-year-old pigs. I'm showing you pictures of the retina on the bottom at 1 year of age, and you can see by already in 1 year of age, these pigs have significant retinal thinning, similar to what you would see in a human patient, and defective ERGs, so their vision is already significantly altered. In the CLN2 pigs, we're seeing very similar things. We've just started doing the ERGs, but in terms of enzyme activity, the enzyme activity for the TPP1 is near 0 or 1%, so significantly lower. And so I think these are really going to be wonderful tools for us as we think about drug screening and continue our drug screening efforts, but also in translating these things into actual human patients. So I will end there with acknowledging the folks in my lab that did the work, along with our collaborators in the Batten disease community. This is really a wonderful research community where the scientists really do work together very closely to kind of push these things together, and then the Batten's foundations that have supported our work over the last couple of years.
Great, thank you very much, Jill. I'm sure we all have lots of questions, but to fit all our talks in, I'm going to encourage you to ask those questions of the researchers during the poster session afterwards, okay? So, as I said, we're now switching gears to a more clinical focus, and we're going to have a series of 3 different presentations from the research group at the University of Rochester. So we're going to, first I'd like to welcome John Mink, who will lead off, and then he will hand on to his colleagues.
Thank you very much, John, for the opportunity, and thank you all for your interest. I'm going to talk today about work done over the past 18 years on natural history studies. I'm primarily going to focus on CLN3 disease, but really use it as a model and as a structure for how we're approaching natural history and other forms of Batten disease. What I'm going to talk about today is really the work of a very talented, wonderful team of investigators. And so many people have contributed to this work. And many of you in the audience have contributed to this work over the years, and we appreciate that more than we can say. So I'm going to talk about prospective approach to natural history studies, and what that means is going forward, evaluating individuals who are affected and seeing how the disease changes over time, which is one approach. It's one that is potentially much more powerful than saying, okay, let's ask how things have been in the past, though that's also an important approach to learning about the disorder. I'm going to talk about what we developed as a kind of a global disease-specific assessment tool, so specific to Batten disease, all forms, and using that to obtain information on how what the characteristics are of Batten disease and how they change over time in a way that we can quantify, in a way that we can actually measure. And I hope you'll get the idea, and I'll talk at the end a little bit about how it really does take a village. It's important, the contributions of many different people from the team, and also the importance of collaboration, as Jill mentioned at the end of her talk, the collaboration among different individuals at different centers focused on rare diseases, particularly Batten disease. Now, some of the data I'm going to show you today, some of the graphs are unpublished, so I'm going to ask you kindly not to photograph those graphs. You can take all the photographs you want of me and of the slides that have words on them. Just don't share the photographs of me. So as I think you've heard, there are some things that present particular challenges to developing therapies for treating the different forms of Batten disease. Most of them have relatively slow progression of symptoms over time. As a parent, it may seem way too fast to you, but as a researcher, it's relatively slow. And the symptoms start during times of developmental change. And we know that during normal development, there are areas where there seems to be a little pause and then a takeoff again. And so we needed to develop a tool that would be usable and powerful in children who are also having developmental changes. And we still don't have clearly established biomarkers. And so my view is what the brain does is behavior, and therefore a good biomarker of brain function is to see what neurologic function is like, including behavior. And then the other thing is we know that different features begin and change over time and over different times in the course of disease. So much I'm going to talk about is data that we have gotten using our tool, the Unified Batten Disease Rating Scale, which was initially drafted by Fred Marshall. I joined him about 6 months into that process. And this is a tool that we initially developed based on what we knew about Batten disease from the literature. And then we went to see a bunch of kids in Toronto in 2002. And we realized that what was in the literature wasn't exactly what we needed to know. And so we added items to our scale based on our experience, and then we subjected it to rigorous testing, statistical testing. Can we do this reliably so we can get the same answer when different people are doing the evaluation? And then continued to modify it, but it's been in relatively stable state now for over 10 years. We have to find people. And so Lisa DeBleek and Amy Verhal have been two of our lead coordinators, though we have other important coordinators. Initially, we established a registry in 2001. We made our first annual visit to the BDSRA family meeting in 2002. And in 2005, we established our Batten Disease Center at the University of Rochester. Over those years, we've worked very closely with the BDSRA, because for this kind of work, you have to find the people who are affected by Batten disease, whether they come to us or whether we go to them, we have to find them. This has a lot of numbers on it. What I'm showing you here really is over the years, starting in 2002, how many individuals, how many affected individuals we've seen each year, how many we've seen in Rochester, and how many we've seen other places, including some home visits. So over the years, we've done a total of 460 evaluations in 183 unique individuals. And that does not include data that we've obtained at this meeting so far. So what you'll see is most of our evaluations are done here at this meeting. And that, I think, is what I want to highlight, is just how important it is to this research to have families who are at this meeting participate in what we do, because, again, we wouldn't learn what we've learned without all of you. This is another way of looking at it. Let's just focus on, well, we can focus here. We've got the individuals with the CLN3 mutation, and then we've got other NCLs. How many evaluations? And there are 3 individuals we've seen really every year for 15 years. And I can't understate how valuable that is, to see the same person over and over, to see how the disease changes, but also it's incredibly valuable to see even children just once, because we learn by comparing across children how the disease varies. About 25% of the individuals that we've evaluated have forms of Batten disease other than CLN3. And so we developed the tool to apply to all forms, and we are gathering data on all forms, though the majority of our information is from CLN3, and that's what I'm talking about in my talk today. So what have we learned? I'm going to tell you a little bit about what I'm going to show you in data. So we know that the core symptoms occur in a relatively characteristic manner that is dependent on time. That although there are individual differences, the big picture is that CLN3 disease is fairly characteristic from person to person. That the symptoms worsen predictably over time. The movement disorder in CLN3 disease is mostly Parkinsonism, which is slowness, stiffness, trouble with balance, as opposed to kind of involuntary movements or myoclonus, though those can occur as well. The epilepsy is of what we consider to be relatively moderate severity, as opposed to some other forms where it's a higher severity. And the cognitive decline, the learning problems and mental processing, declines relatively early and then slowly progresses over time, and that's what Heather Adams is going to talk about in some of her talk, which will follow mine. And then the combination of these features really work together to lead to the loss of functional abilities. So here's some data. I'm not going to go through all the details, but I want to point to a couple of things. So on average, vision loss comes first, and then behavioral and cognitive problems, and then sleep problems, and then seizures, and then movement problems, and then ultimately trouble with feeding, primarily with swallowing. There are some slight differences between boys and girls. On average, girls develop symptoms about a year later than boys, but it seems to move a little bit faster in girls, so that on average, girls lose their ability to feed independently and develop trouble with walking a little bit earlier than do boys. Another way to look at this, and this is the work that Margot Maston's been doing with us this summer, has been to look at the order of onset. When do the symptoms start? Regardless of what the symptoms are, at what age does the first symptom occur? At what age does the second major symptom occur? Third, fourth, fifth, etc. And so what I'm showing you here is the age along this axis and the order of symptom onset. Each dot here is the average across all the individuals that we've evaluated. And the bars here are what we call the 95% confidence interval. That means that 95% of the individuals fall within that range. And that, if you look here, you'll see those are really small. So this tells us this is a fairly homogeneous process, meaning across individuals it's pretty much similar. And on average is about 2 years from one major symptom to the next, to the next, to the next. So we can predict fairly well what the rate of progression is going to be, and that can be very useful potentially for design of clinical trials, and what do we use as measures to see if that predictable order has changed. Using our physical rating scale, which includes things like talking and walking and moving the limbs, this is a scale where the severity is showed here on this y-axis, the vertical axis, 0 being no symptoms, and our maximum possible is about 112, and so high numbers are worse, low numbers are better. This is based on data collected over the years of 120 children with proven CLN3 mutations. And what you can see in the first few years, it's a little bit flat, but then after that, it's a fairly regular progression. And again, these error bars are very small. So it tells you that we can use this to both assess and potentially predict how the disease is going to progress based on the physical aspects over the course of here 20 years. What about seizures? And this is some updated data, but work that was published a few years ago by Erica Augustine, who will be speaking third in our group about different aspects of our work and what they mean for clinical trial development. But here too, this shows that overall, the seizures, high numbers are bad, low numbers are good. Seizures get slowly, progressively worse over the course of the disease. Most seizures in CLN3 are what we call generalized tonic-clonic or grand mal seizures. And the seizure frequency, how often they occur, is compared to other children with epilepsy, is what we consider kind of moderate. What about functional capability? How do the children do in school? What kinds of games can they do? What kind of chores can they do? What self-care can they do? And here, again, 0 is completely independent. High numbers are worsening loss of independence. And again, you can see a pretty steady and fairly homogeneous progression over age. So we've got these different ways to characterize the disorder, and we can put them together in different ways to get a good picture of the disease. And that picture of the disease is really important for then developing and evaluating clinical trials, because we want to know how treated individuals compare to what we would have predicted without treatment. So in 2002, we started with the idea that maybe one day there might be therapies, but it was a fairly distant hope. And there were very few models for studying rare disease in human beings, particularly in children. Now, 17 years later, really going on 18 years later, we have an approved therapy for CLN2 disease. And there are clinical trials either in process or development for many forms of NCL. And our rating scale work, our natural history work, is being incorporated in many of these trials as the way to compare how the treatment is affecting progression compared to what we would have expected without it. So we've developed this tool. We've been able to use it to quantify progression, particularly in CLN3 disease. We've been able to generate a very large data set of information for testing hypotheses as they come along. Some of those hypotheses we've gotten from parents. And we've gotten those ideas from parents where we can then go back to our data and say, I wonder if. And sometimes we can support those, and sometimes we don't support them, but what we learn from you is critically important to what we do. This can be used as an outcome measure for clinical trials, and again, Erica will be talking about that. And a model, I think really a model for rare disease research in neurodegenerative diseases of children in all forms of Batten disease, but also in other rare diseases. This is pretty small type, but this is our team, and the team over the years has evolved. In yellow here are the individuals who are here at this meeting currently, and I hope you will all have an opportunity to meet at least some of us. One of the points of this slide is it's multidisciplinary. So we've got neurologists and neuropsychologists and statisticians and research coordinators who are really the glue that hold us together. And one of the things I'm proudest of is each summer to the BDSRA meeting, we take 2 students with us. And those students get experience with Batten disease. Some of these have gone on to become clinicians who have direct involvement in caring for people with Batten disease, but they've all taken that experience and their knowledge to inform the development of their careers going forward. And then, of course, you can't do anything without money. And these are organizations and individuals, organizations that have supported our work, and then collaborators. And I don't have time to talk about all of our collaborators, but we have really important collaborations that are ongoing or developing with colleagues at the NIH and with the Mass General Hospital Group and with our colleagues at the University of Hamburg. And I really consider the BDSRA to be an important collaborator through all of this. I'm going to move on, just finish with some pictures of our group. We need to update this from this conference, but this has really become a passion for us, and it's wonderful to be able to present to you the work we've done over the years. I'm going to now introduce Heather Adams, who's the neuropsychologist in our group, to talk about her work, and then she'll introduce Eric Augustine to talk about her work. Thank you.
Does this work? Yeah. Great. Which one is the pointer? Okay. Hello. I might just move around a bit. I'm a bit restless. So I'm also going to talk about progression and the sort of arc of time in studying a disease over many years. I was asked to talk about cognitive processing, in particular memory and language, although we have data on lots of other areas on cognitive processing in CLN3. And I have a little brief mention about adaptive functioning as well. And these are my disclosures. And again, I also have some slides that present data that are not yet published, so I'll ask you to kindly not take photographs of those. I want to echo what John said as he closed his talk, that our partnership with the BDSRA and our partnership with you as family members of affected individuals is really critical. And I think 80% or more of the data that I'm presenting and that John has presented has come out of these meetings and has come out of the time that you and your affected children have given to us to help us learn about these diseases. So we thank you very much, and we want you to understand that you here sitting in this room and many who've come before you are the reason that we can share these data with you. So there are a few challenges when you assess symptoms in the NCLs, and this is true for all NCLs, not just CLN3 disease. First of all, they're rare diseases, so there are very few opportunities to learn about the pattern of change if you only have a few individuals and they each have a slightly different course, or they have different genetic variants or different mutations. It's really hard to make broad statements to say on average children with CLN3 or CLN1 or CLN6 look like this if you just have few instances of the diagnosis. And so we try to cope with that by studying as many individuals as possible over as much time as possible so that we can develop that body of knowledge. Also in children, there's very much, of course, an assumption of development, not regression. Children develop and acquire skills over time. They grow taller, they learn more, they're capable of doing more, they build their vocabulary and their skills across all areas. And many of the assessment tools that we use and that are available for us for assessment of cognition and behavior and adaptive skills in other areas are really built upon that assumption of development. And so what you can then experience when you're evaluating children who perhaps lose skills over time is what's called a floor effect of the measure, where the tests and the tools that are available for us to use aren't sensitive enough at that lower end of ability to really understand what children still can do, because they're expecting children to be able to do more based on their chronological age. And finally, I think the majority of the measures that are available for us to use don't consider that significant and evolving change in physical disease burden. So we know that individuals with NCLs experience vision loss, changes in their ability to speak, to walk, and to talk. They do develop dementia, and they can have challenging behaviors. And so to find one assessment tool that encompasses all of those symptoms and then doesn't get distracted by other non-relevant behaviors or cognitive skills is challenging. I think the Unified Batten Disease Rating Scale, which John presented on, is a great example of how we move forward with developing something that's disease-specific for the NCLs that really focuses in on this multiplex set of symptoms for these different disorders. So another thing that comes up, and particularly for assessment of cognition and behavior and adaptive function, is the question of how do we interpret the scores that we get. So if we take this hypothetical example, we have, say, a test with 25 questions on it, and if you can get 1 point for each question that you get correct, the maximum possible raw score you can get on this test is 25 points. Well, let's say you have a bunch of children from 2 years old up to about 16 years old, and they all get 15 questions right. Well, what does this mean? Does this mean that the little 2-year-old here is a genius? Or does it mean that the oldest kid is maybe lagging behind her peers? Because you might expect, for example, if it's a test of vocabulary or spelling or memory or even some physical skill, that older kids should be able to do more on a test if they're developing at an age-appropriate level. And so that score of 15 in isolation doesn't really tell us much about where that kid is in relation to their same-age peers. However, if we follow a kid over time and we look at that change in their own individual raw score, that score then becomes meaningful for that child. So perhaps that toddler at age 2 is getting a score of 15, and then a couple of years later, they're getting a score of 17, and then they're getting a score of 20 or 22. What we would find is that we are seeing some demonstration that that child is developing and acquiring some knowledge and skills over time. Now, we don't necessarily know if the rate of development is similar to other typically developing children, and for that we have what are called age-adjusted scores, which is a way to sort of statistically adjust scores, these raw scores, at any age of a child to understand where they are in relation to their same age peers. And this just summarizes what I told you. So I got ahead of my slides. So in our study of CLN3 disease particularly, although we have used this model with other NCLs as well, these next few slides will show you some particular results of these raw score changes in individual children over time. We have now studied probably over 80 children who have genetic confirmation of CLN3 for the neuropsychological testing portion of our study in particular. And over this approximately 15-year period from 2003 when I started to 2018, we had 180 evaluations in many different children, in these 77 children. And some of these children had had as many as 9, 10, 11, 12, 13 assessments. Many of them had at least 2, between 2 and 4. So there's a couple of things on this slide I want to point out to you. Like Dr. Mink's slides, we have these points on the graph which show you sort of an average at any point in time. We have age across the bottom, the x-axis, so that's the horizontal axis there. And then on the vertical axis of both these graphics is the number of details that a child's able to remember after reading a story to them. So we verbally read 2 short stories. They're only a couple of paragraphs long, and then immediately after reading the story, we ask them to tell us as many details of the story as they can remember. And then about 20 minutes later in the evaluation, we ask them to tell us how many they still can remember. So it's immediate recall and it's delayed recall. And both of these are tests of their independent ability to remember the story details without any help or tips or prompts or cues or hints. How much can you tell us about the story? And what we can see is that we have, on average, for children who are being tested for the immediate recall, that you can see that even though perhaps if we looked at the age-adjusted scores, they might not be remembering as much as other children their age, in these graphics you can see that there still is some increase in their ability to learn and retain information, and that seems to be retained for longer for the immediate recall portion. If you look at the delayed recall on the right-hand side, that it begins to drop off and decline a little earlier in the age span of the children affected by CLN3. So I think that has bearing for understanding something about the disease, and also I think is information that then can be translated over to real-world settings like schools, where we can think about what's our window of opportunity for teaching children, and what sort of methods should we use to assess what they can learn and remember, and when should we be checking in with them about their learning. Recognition of story details. Remember I said that these two have to do with independently how much you can remember without any hints or tips or cues. This is different. Now we say to children, okay, we're going to ask you some multiple choice questions about these stories. Was the girl's name in the story Emma, or was it Jane? And you tell us if you think that you can remember the name of the girl in the story. And here what you see actually is not only is there an improvement in that over time, but also that's retained for much longer. So what that suggests to us is that children are remembering, they are learning information. I will say that it's not at the same level of typically developing children, because these raw scores of the number of details they're recalling is lower than age expected. But nonetheless, relative to their own self and their own progression over time, there's still some capacity for retaining information, and that persists for longer over the course of the disease. That suggests to us that there is more of an issue with the strategy for how you independently recall information, and it's not necessarily that they're forgetting, it's perhaps that what the details are that they have learned, it's harder for them to kind of independently access that information. But if we give them strategies and support to do so, like multiple choice questions, they can demonstrate what they truly do know. And again, that has bearing for education, as well as for just scientifically understanding things about this disease. I have just the one slide on language. This also has to do with sort of the strategies for learning and strategies for doing tasks. We have a test that's very commonly used in neuropsychology called verbal fluency, where we ask individuals to rapidly tell us as quickly as possible, within a minute, as many words as they can think of that are either types of a certain thing, like a category. So tell me as many different types of trees that you can think of. Or we ask them to tell us as many different types of words that begin with a certain letter of the alphabet. And both of those are tasks that really both rely upon verbal ability and vocabulary knowledge, but also those strategies and executive skills for accessing information within a certain category, remembering the rule of the test and not deviating from it and getting distracted. So if I ask you to tell me words that start with the letter G, you don't tell me words that start with the letter P, for example. And being able to kind of develop a strategy for generating as many words as possible. And what we see for CLN3 disease, again looking at raw scores, so on the bottom is the age of the child, and on this side is the total number of words recalled, is that being able to kind of handle the categories is retained for longer and doesn't decline as dramatically. And again, that just tells us something about both verbal ability and executive strategies for using language. So I just want to very quickly shift gears, because I know I'm short on time, to talk about adaptive function, because that's very important as well, not just for understanding the phenotype or the characteristics of the disease, but for real-world, everyday function. Adaptive function broadly could be defined as the skills, conceptual skills, social skills, practical skills that a person has to handle the common demands in everyday life. Being able to use the toilet or communicate that you need to use the toilet. Being able to feed yourself. Being able to have a social interaction with another person. These are all adaptive skills. For many years, we collected data on adaptive skills using a very short form called the Scales of Independent Behavior. It was a short form for the visually impaired, so it only included questions that didn't require vision on the part of the person being assessed. And unlike the neuropsych data, which is based on a performance outcome of the child themselves completing the test, the CIBR data are based on parent reports. So these are parents' keen, astute observations of their child in everyday life. And what we can see is there are these age-related changes in their broad independence on the measure. And that you start to see it's really by around age 10, 11, 12 that there's an initial sort of drop in skills, and then between about age 12 to 15 or 16, there's a gradual decline, and then after that, adaptive function skills start to drop off more rapidly on average for a group of children. And we think that this probably is very closely associated with the physical changes in the disease as well. So some take-home messages. Over time, cognitive and adaptive function abilities level off, and then they decline, and we want to consider this when planning curriculum or training for acquisition of skills that children might need later in life. If you went to the JNCL and Education session yesterday that was presented by a bunch of people, we talked about the fact that there really is this window of opportunity for learning and developing skills for children affected by the NCLs, whether it's CLN3 or any other, and you really want to sort of jump in when children are still acquiring or when they're sort of leveling off in their skills rather than waiting for declines to kick in, so that you can really build up a bank of skills that children can then use later in life. Although I've talked today mostly about the raw scores, it is useful to compare to age-based performance, and that is the data here is age-based, and that's to sort of establish expectations or to help you access services. Often, in order to access services, individuals need to kind of cross a threshold for a score compared to same age peers. Although I've presented data today only on CLN3, we have been able to use this approach with many NCL types, and we're really building our catalog of data and information about many different NCL types to be able to generate those same sorts of graphs going forward. And finally, I'll end by really just emphasizing that that partnership with the BDSRA is really critical, because again, what I've presented here today is really, I would say, by and large, a function of being at these meetings and being permitted generously by the BDSRA and families to be at these meetings and participate with you in the research. So again, this is very similar to John's slide, but I'm sure none of the people here would mind if I just simply thank the families. So thank you again. And I'll now introduce Dr. Augustine. She is the lead in our group for clinical trial development, and she's going to talk a bit about that and some other things.
We're about to run out of battery.
Started anyway. Sounds like our computer out here is going to run out of batteries. And if we do, then we'll move to Q&A. So either way, we will make productive use of our time. Oh, we've totally run out of batteries. So, oh, we're back. Thank you. So we've been talking about natural history, and I'd like to speak a little bit about how we moved to trials. And along the way, hopefully I can try and bring together a lot of the concepts and topics that we've discussed this morning, all the way from the preclinical work and animal models to really where we all want to go and where we want to be in developing new treatments. We've hinted at this idea and talked about some of these concepts, but I thought we'd just start by taking a minute to visit what therapy development typically looks like. For some of you, these are very familiar kinds of concepts. For others, these are a little bit newer, and I can talk through this even without slides there. So we really begin with trying to understand the disease, and we begin with thinking about preclinical models, animal models, where and how can we learn about, well, what causes the disease, and how do we identify what the processes are that take place in the disease? And over the last 25 years or so, in some ways we've come very far in understanding that a little bit more from animals, and certainly being able to now understand that Batten disease is not one disease. It's many, many diseases, each with a distinct genetic cause. Recognizing that has helped us to have a leap forward in how we think about therapies. And along with that, the development of therapies like gene replacement therapy and other strategies that David Pierce talked about helps us to be able to move forward with clinical trials even before we can really even fully understand what these proteins do. We still are able to continue forward. So that's that first piece, really understanding the disease. The next piece is trying to figure out, well, what might we target? What might be reasonable treatment options? And to be able to do that, again, takes some being able to identify these therapy targets, and then you move that into trying to understand as best as you can, well, what might be the right dose for that approach? What might be the best route of administration for that approach? And how safe is there any particular possible treatment, and how well does any possible treatment work? And you do all of that work in animal models. And when you really have a good sense, okay, I have a concept about what might be a good dose, I have a concept about what safety and benefit might look like, you then move into human studies. And those human studies, sometimes that's one trial. So many of you are familiar with the idea that for sarlipanase alpha, that was one trial. That's relatively unusual. It's usually trials plural. And you're trying to answer all of those same questions that you were looking at in animals. You want to try and answer those questions in people. How do I give it? Where do I give it? What's the right dose? Is it safe? And does it work? And so when we think about going from, maybe I'll move to a microphone. Can everyone hear me? Is that still working? Good. Okay. And so there's a password up here of some sort. But when we go into those human trials and you're working through those questions, I think as we go from natural history to trials, we think a lot about that idea of benefit and what does it mean to have benefit. And when you get to a good place, you then ideally have great safety data, you have great efficacy data, and you can go to the FDA and say, we've done all the work, this really looks promising, let's approve that so not only people in the trial can receive this, but again, anyone with the condition, with the right component of things can actually look at that too. Thank you. Or maybe not so much. But the thing that I want to emphasize here before we move on is that we use a lot of terminology, and we talk about research, we talk about clinical research, we may speak about clinical trials, and sometimes people use the word treatment trials. And the thing that I want you to take away is that in this therapy process, our goal, our hope is that we are truly creating new treatments, but we don't know yet. And so we have lots of hope that these are treatments in the sense that they are safe and that they will work. But really, until we get to that point, that part way over here, or maybe I'll use this one since everyone seems to be on this side. Until we get way over here to approval, it's not a treatment yet. We're still learning. And so again, our hope is that all of the things, all of the promise and hope that we have about new compounds is that they will work and that they will have benefit and they will make a really important impact on lives. But sometimes things don't work. And so it's important not to necessarily equate trials with treatment. They might be treatments, but we do the trials to make sure that we can find out and understand that really well. So what about rare diseases more broadly? You know, again, why is it difficult? Why is it challenging? We know that there's a need. We clearly need treatments. We understand where the gene where the etiology comes from. We talk about these gene therapies. You can get it there in mice or in pigs or in dogs. And why doesn't that just mean that things will definitely work for people? Or where are the challenges in trying to understand how we get from mice to children? Well, we're talking about very rare diseases, and so then there are some clear elements that make sense here. It's rare, so not a lot of people have the disease. Usually when we're talking about trials, we go from talking about small trials in a few people, sometimes to trials in hundreds or thousands of people. We don't have hundreds or thousands of people who have Batten disease, so we have to be creative about how we think about what a trial looks like. Many of you have asked questions about different symptoms, and why siblings might have different symptoms, or why your child's symptoms are very different from someone else's symptoms. And scientifically, we're learning a lot more about these concepts of what we might call genetic heterogeneity or phenotypic pleiotropy, all meaning that different genes that cause different diseases sometimes cause symptoms that actually all look quite similar, even though they're different diseases. And sometimes one gene can cause different kinds of symptoms, and we need to learn a lot more about that. And how do we think about conducting trials when people are really different from one another and their symptoms may be different from one another? A lot of the groups today spend a lot of time thinking about these areas where there are arrows. So what is natural history? What are the important symptoms? How do symptoms unfold? Jill Weimer talked about an approach to really trying to identify and validate biomarkers or objective biochemical measures of disease, whether that's diagnosis or progression or symptom severity. How do we think about that in a biomarker context? And then even though we talk about natural history, it's actually a different thing from a clinical endpoint. And so that's where we want to spend a little time today. So natural history, the work that John Mink and Heather Adams spoke about, it's been really helpful in trying to get some insights into understanding the disease and the way that, or the diseases and the way that they affect groups of people. We've been able to understand, and these are data from CLN3, well, how do symptoms unfold? And these curves represent one symptom and then a different symptom type and a different symptom type. What's the order? What does that look like? We've been able to do things like quantify progression. Can we put numbers to how things get worse? And this is sort of an image that looks at motor symptoms and being able to track how that changes over time. And with those natural history data, we've even been able to ask some scientific questions. Well, are girls different than boys? Are there specific predictors of what might happen in the future? And so we've learned a lot just about, again, what are the important symptoms? It actually has helped us a lot. The research has helped us a lot as doctors to be able to help families and patients know what to expect, what might be on the horizon. There are slightly different questions, though, when it comes to trials. And in the end, what we really want to know, again, we talked about safety. Is any particular potential treatment safe? We also want to know, is any particular potential treatment effective? Does it work? And when you're looking to prove that something works, in the end, you want regulatory agencies to agree with you that it works. And so we think about that in what we call clinically meaningful effects. So it's not just that we can make a number move or that something is slightly different. We want to be able to clearly show that a potential treatment has an effect on how somebody feels, on how they function, or how they survive. And so different ways that might show up, well, maybe there are fewer symptoms. Maybe people can do more things, or they can do things for longer. Hopefully people live longer. Hopefully they feel better and have fewer complications. And again, longer, better, higher quality of life. And these are the kinds of things that we're looking for when we're talking about whether or not a treatment works. So how do we get ready for clinical trials? So to bring this back to the very beginning, David Pierce talked about many compounds in the pipeline, some of which are being tested already, some of which are close to being ready to test, and then others that are going to take a little bit longer to get there to clinical trials. And along the way, it's really important that we partner well with our folks who are conducting preclinical work in terms of new discovery, therapeutic discovery, and new targets, and also that each time that we go through this trial process, not only are we ideally identifying new treatments, but that each time we do it better and better, and that we're more efficient and we're faster and faster. When you think about something like high blood pressure, it's clearly different than high blood pressure, what we're talking about here. But when you think about high blood pressure, it affects millions of people. And there have been decades and decades of trials and many, many compounds. There wasn't one trial that created one drug that solved the problem of high blood pressure forever. Each time, there's a need to get things better and better. And even though what we're dealing with is clearly very, very serious, and each step is really important, we know that one trial won't be the answer. It'll be an important step, ideally, but there will be more steps after that. So clinically, we want to make sure that we keep developing the information that helps us do it better and better and get to those answers and those new treatments faster and faster. So what does it mean to be ready for trials, and what are we focused on? Well, some of that has to do with collaboration, sharing information, sharing data, developing common approaches, standardizing our data elements. So the way that I collect information in Rochester is the same way that information is collected at Nationwide Children's, is the same as how they do it at the NIH, it's the same as the way they do it in Hamburg. And being able to come to agreement about how we do that, to set up processes for how we share data so that your data at the time that patients and families commit is as meaningful as possible is something that's really important. We want to validate outcome measures. We want to know that we not only have natural history, but again, we have things that can be used in trials, that we've trained people to be able to use those particular tools. We have biomarkers that are valid for disease progression, and that sometimes that means that we're actually identifying new ways to do trials and to think about new methods for trials in rare diseases. And all along the way, for all of those elements, it still remains important that we understand natural history, just slightly distinct from these particular goals. So in terms of our trial readiness effort, I'll take a moment to talk about just two particular NCLs, but again, all of this information really applies for all NCLs. And so to think about CLN3 disease, where we have really solid knowledge about natural history, we're really at a good point to move forward to thinking about trial readiness and to build on that further. We just started a new study that we launched in May, supported by the NIH in collaboration with the University of Hamburg. These need to be collaborative efforts, multi-center, multinational efforts. And we are looking to enroll 35 children and young adults who are impacted by CLN3 disease to be followed and enrolled in the study for up to 3 years to, again, really think about systematic evaluation. And I'll talk a bit about that and what the goals are. We will learn about natural history, but we have other kinds of specific goals as well, developing those trial outcomes. We had a meeting with the FDA. The FDA, I think many of us are finding, are really important and critical partners in moving forward. So even when they're not here in the room, they're sort of in the background of what all of us are doing, whether that's discovering a new therapy and trying to bring it forward, or the work that we do, again, and being ready for that. And in that, we spoke a lot about outcome measures and assessments that are available and used in children very broadly, and then also specific kinds of tools that we think about for children and young adults who have NCL disorders. And we received guidance that helped us think about what's that next approach in being ready for trials, doing things better and better with respect to this kind of global disease outcome. How do we think about what the right approach is to the important content within our rating scale, the UBDRS that John spoke about? How do we make sure that our scoring algorithms are correct? How do we make sure that we have the most statistically robust approaches, the most sound, specific approaches that help us to use these tools in very small groups and still have reliable information? What's our best approach to scoring? How do we highlight those elements of feels, functions, and survives that I mentioned earlier? And then how do we make sure that we have approaches that can happen across the nation, across the world, in terms of training processes for using this particular tool? And then we'll apply that to other kinds of tools as well. So in terms of some of those objectives, some of that has to do with these measures of disease severity and looking at our UBDRS to take a very deep statistical analysis approach to figure out how does this measure work the best. We also want to look in a very detailed manner at different symptoms. So you heard Dr. Adams speaking about cognitive assessment, and some of you may have said, well, Dr. Adams is talking about storytelling and remembering story and generating words, but my child's not speaking anymore. I know that they're still understanding what's happening, but how do they convey what's going on and what works? So for some of these approaches, we want to look at lots of different strategies to try to figure out, well, how do we actually still convey what a child might know or try to assess what a child might know, even when they can't talk to us anymore? Some of you have had the experience of going to the ophthalmologist, and the usual and typical kinds of tests that take place, they're hard to do. So we want to be able to say, okay, well, certainly we want to think about things like visual acuity. You might talk about someone being 20/20 or 20/40 or 2400. Well, what do things look like under a microscope when we look at the eye? What do things look like electrically when we look at the eye? And what are all of the ways that we can evaluate how the eyes are working? And if we take that battery of tests, just looking at vision, what are the approaches that work the best over a variety of ages, over a variety of children with different abilities? And so these are things that we'll learn about that as well. And same kind of deep dive into epilepsy and how we best assess epilepsy. You heard one of the first early speakers talking about a catwalk for mice. Well, we're going to have children walk the catwalk too, so that we can actually have better translation of our preclinical evidence into patients as well, and vice versa. What can we look at and learn about how quickly gait might happen? What are the ways that we can look at those footfalls or footsteps and understand what can we learn and quantify about gait analysis, and to be able to translate that very directly and very clearly from children, again, to mice and back. We talk about disease staging. Dr. Adams mentioned adaptive function. These are things that are going to help us think about large milestones in disease and everyday kinds of activities and how they're impacted and how we track that over time. These are some of the elements that, again, are standard assessments that one might expect to see in trials that haven't been rigorously and detailed, in a detailed way, assessed in children who have NCL disorders. And so we're going to take that deep dive so we have that information available. We think about biomarkers, clinical biomarkers and neuroimaging. This is work that has been spearheaded by Dr. Schulz's group at the University of Hamburg. Our collaborative trial is really an exchange of expertise of outcome assessments on the University of Rochester side and neuroimaging depth of experience at the University of Hamburg, and we will use each other's sites as an opportunity to ideally replicate the data that each has found, so that we can think about what does disease progression look like when we're taking a look at images or pictures, how do we translate that into a numerical progression as well, and then we can use the other data that we have to understand better what does that actually mean. And then we talked a little bit about training as well. We need to have ways that we can, across the world, be able to have standard procedures in place so that we can all approach assessment in the same way. And how can we identify disease milestones? Are there specific points in time that we can think about that might be events that are important for clinical trials? Onset of seizure, onset of vision loss, time where there might be a need for feeding support. Can we really get very specific about those, and can we use those in trials? So although what I've mentioned has to do with CLN1 and CLN3 disease, these are factors that are true for all NCLs. And every gain in thinking about the approach to any one NCL really does something that can have impact across all NCLs. And the shared experience across the Batten Centers of Excellence advances each time we take a deep dive into any one NCL. So we're applying these same kind of strategies, not just for CLN1 and CLN3, but actually to all forms of NCL, and for each new approach to think about, okay, what are we trying to accomplish with respect to trials, and design the studies in that way. The very last thing that I want to leave you with is that while the researchers have their specific clinical trial readiness goals and the things that they need to do to prepare for trials, I would say there also are some things that for families, as you think about clinical trial readiness, that you may want to consider as well. So you are in a position to be very informed families as it comes to research, and to be asking the questions to researchers about, what are you studying? Why are you studying it? What do you think this particular new therapy or new potential therapy might do? Who is eligible for the study? What does my commitment look like if I'm to enter a trial? These are all questions that you'll want to be asking. And then think about whether or not you are able and have the time to participate in some of the work preparatory to the clinical trials that really helps them to be better, more important, more efficient, more powerful. And there are lots of ways to be able to participate in trials, whether that's in person, traveling to a site, or the kinds of things that you can do remotely from home, even through surveys. So think about whether you may be able to contribute to some of those. I encourage you to come and talk to us. I know we're out of time for Q&A. I think we will all stay after, but I encourage you to come to talk to our University of Rochester group and all of the other research groups. We're here to answer questions. We're certainly here to conduct research, but we're also here to get to know all of you. So even if you have no interest in participating in research, we still want to meet you and engage with you, and so hopefully you'll come and talk to us. And we'll stay after for questions. Thank you everyone for your time throughout all of this.